Shalin Naik. All right, let me start again. Thrilled to have as our speaker, Dr. Shalin Naik from WeHi. Shalin is the Walter and Eliza Hall Institute. Shalin is a graduate of the University of Queensland, microbiology and biochemistry. Did his PhD with Ken Shortman on dendritic cell development at WeHi. And right then, he got interested in single cell tracking and fate determination very early. So that was in 2006. So way before the rest of the world got onto this. And wanted to pursue the cellular barcoding. So he did his postdoc with Ton Schumacher at the Netherlands Cancer Institute, where he traced the single cell output of hematopoietic stem cells in vivo, really pioneering work there. And then he returned to WeHi in 2013, was lab head in the immunology division. And his lab at WeHi uses single cell and clonal systems biology to learn about immunology, cancer, and development, all biological process of Deep Institute to the GIS and other institutes here. He's made major contributions to understanding hematopoietic stem and progenitor cell heterogeneity using genome technologies that we'd love to hear about, really cutting edge tools. And he set up the single cell open research endeavor, SCORE, at WeHi. And some of you may know he's also something of a celebrity and a TV host of popular science and a podcast host. So maybe one day we'll air one of those videos as well. Shalin, welcome.
Thanks very much. I really appreciate the invitation and for all of you to join, even though remotely. So yeah, I'd love to tell you about the work we're doing in clonal biology in development, hematopoiesis, and cancer. And so this slide is actually data from a paper I'll talk briefly about at the end, where we studied triple negative breast cancer in multiple parts of the same tumor. And as you can see, each dot is a clone and the color corresponds to different clones, and the size is the relative clonal frequency in that tissue. And as you can see, there's so much diversity in clonal composition that we really need an additional lens at the clonal level to understand biology as a whole. Single cell RNA-seq has got us a long way there, but now we need to bring in the clonal information. And so that's something we've been working on for many years. Okay, so you have a population of 10 cells. Let's say they're stem cells or naive T cells, or any cell that can divide to give rise to 2 or more cells. That's my definition of a clone today. It's not genomic heterogeneity, it's just single cells. Now, if you put those cells in culture or in vivo, and they make red, blue, and green cells, there could be many possibilities. And 3 extreme versions are that each cell makes all 3 cell types in equal proportion and number. Another situation where a subset produce red, a subset blue, and a subset green. Or there might be heterogeneity in clone size and composition. And actually, there's an infinite number of possibilities of how, these 10 cells could give rise to, a certain number of red, blue, and green cells. So really, our lab tries to understand what are the patterns of clonal production and what are the molecular programs that control it in all areas of biology. So broadly speaking, we're interested in hematopoiesis, which is blood cell generation, regeneration, dendritic cell development, which is a type of immune cell, part of the hematopoietic landscape, and clonal cancer biology to hopefully identify novel targets for cancer therapy. And the central, tenet of how our lab operates is the integration of biology and medicine with computational biology and technology. And we really try and make sure we are, leading the way in each of those. It means we might not be the experts in any one of those, but I think the integration of them really makes a big difference. And so let's talk about hematopoiesis for a minute. So you might have seen something like this in a textbook or a talk. This ball and stick diagram, where on the left here, I'm just going to put laser pointer on. Here's a long-term stem cell, which gives rise to short-term stem cells, multipotent progenitors, which then branch out into the lymphoid pathways that make T and B cells, and the myeloid pathways that make macrophage, eosinophils, megs, erythroid. and dendritic cells kind of come from both. Now, the implicit assumption in these diagrams, if you zoom in just here, and then we do it over here, let's take the multipotent progenitor. The implicit assumption is that every cell within that population is able to give rise to every downstream cell type, right? But is that really the case? And that was the subject of my postdoc with Ton Schumacher. And instead of being able to analyze single cells by injecting them into individual mice, as in this diagram, we thought, wouldn't it be great if we could barcode the cells differentially and then inject them into the one mouse? And so cellular barcoding existed when I joined the lab, but then we converted to kind of a next gen version that used Illumina and high throughput and everything. It was all microarrays before that. Very powerful, but limited in its scope and quantitation. But now we have this great technique. It took 4 years to develop. It took 4 years to develop, 1 year to do the experiments and 1 year to do the analysis. But anyway, long story short, the principle is that you have a lentivirus pool. Each lentivirus has a different DNA barcode. You infect stem and progenitor cells of interest with this library. They're integrated into the DNA and inherited by the progeny cells. So you inject those cells into a mouse and they divide and differentiate as they ordinarily would. And at the end of the experiment, you take cell type A and cell type B, which are your cell types of interest, and you do barcode PCR and sequencing. If a barcode is only in one cell type, like this blue one, it's indicative.
Shalin, sorry to interrupt you. There's a couple of people messaging that they're seeing the speaker in the main screen and your slides on the side screen. I don't know if this is a problem. Their issue is, Lynn, can you check on this? Or Winnie?
It's fine on my side. I'm seeing, I'm seeing the slide on the main screen.
Okay, great. Me too. Yeah. So maybe it's just on the other side. So if
sorry, I realized I was on mute. They have to click the top right under view, so we won't be able to change it for them. They have to adjust it on their computer.
Okay. Thank you, Lynn. Sorry for the interruption.
All right. And so this barcode 2, if it's present in both cell types, it means it came from a bipotent progenitor. And barcode 3 was only in this cell type, came from monopotent or mono outcome B only progenitor. Anyway, so on and so forth. We tested whether multipotent progenitors were actually multipotent in vivo, and the answer was no, they're not. So here, the percentage of LMPPs that could generate all different cell types, where you would expect it to be 100% if they were multipotent, was not the case. It was less than 5%. And instead, a proportion of progenitors was either B cell biased or dendritic cell biased or myeloid biased or combinations of the two. And we also demonstrated in clone splitting experiments into different mice that this was an intrinsic property of clones. So we saw the same barcode end up in the same cell type in 2 different mice. That tells you it was intrinsically programmed at the time of injection and probably programmed in that cell prior to isolation in the first place. So this is how you can identify the patterns of lineage bias. Okay, so this data, along with many, many other people, including a lot of single-cell RNA-seq studies and pseudo-time trajectories and whatnot, have given rise to this concept, not of the ball and stick model, but of the continuous model. The idea being that when we use flow cytometry FACS to draw a gate around a cell type that we think is pure, actually, there's a lot of heterogeneity within that box, and we don't yet have the tools to disentangle these in terms of lineage bias, so that within these common myeloid progenitors, this subset of cells is biased towards basophils, whereas this subset of cells is biased towards monocytes, for example. And so barcoding helps you untangle the types of fates that you have. Single-cell RNA-seq helps you identify the molecular nature of these. And we wanted to zoom in to understand if we could subset these a bit more using prospective markers of isolation. And like many people, we did straight up single-cell RNA-seq of a population region of interest to understand if there was heterogeneity. We did it with plate-based sorting to get some FACS markers as well. So in this heat map, you can see the FACS marker expression of single cells, which are in columns, but also a similarity to ImGen reference data set. And to cut a long story short, we identified this novel cluster of lymphoid-like cells that were not lymphoid progenitors, that were IL-7 receptor negative. They were a subset of MPPs, and they were higher for FLT3 expression, c-KIT and SCAR1, but also a common lymphoid progenitor signature. And what was quite notable about them was their expression level of DAC1. And as you can see, there's a force-directed layout of that same data. And so this little, outcrop here of cells, which we call the lymphoid prime progenitors, expresses these two lymphoid genes, is low for DAC1 expression, high for FLT3. Long story short, we use these two markers to sort them to purity, test that they were lymphoid biased and could not generate the classic myeloid cells. And so we were able to subset the MPPs into the lymphoid bias. So I showed you that barcoding data earlier, where a subset of progenitors made B cells. Now we know how to get those B cell only progenitors. All right, so that's one way to identify, novel regulators of priming, lineage priming. But is there another way? And that relates to the second theme of our lab, which is dendritic cell development. So as a one-slide introduction to what dendritic cells are, they're a type of white blood cell in the immune system. They sense pathogen and cancer, and they educate T cells through a cognate peptide MHC TCR reaction. And for the purpose of this talk, there's three subtypes of DCs. Conventional DC type 1 that targets intracellular pathogens, CDC2 that extracellular pathogens and antibody responses, and plasmacytoids that are important for type 1 interferon and antiviral responses. And importantly, CDC1 is really important for anti-tumor immunity and immunotherapy, and increasingly the target of new biotech startups and preclinical and clinical trials. And I'd just like to shout out to my PhD supervisor, Ken Shortman, who actually discovered these 30 years ago to this day at WeHi, where I'm sitting. And he called them CD8 positive dendritic cells at the time. So for many years, he was accused of stamp collecting. But now this is one of the hottest cell types in immunotherapy at the moment. So it just goes to show that sometimes you need to persevere with interesting observations rather than application-driven science. So what controls the fate of the DC subtypes from multipotent progenitors? So in 2005, during my PhD, I came up with a culture system where you take bone marrow from a mouse, you culture it with FLT3 ligand, and you can generate the three DC subtypes, CDC1, 2, and PDC. It also allows you to drop in a single GFP labeled cell in that culture and track its clonal output. And there, to our surprise, where we seeded single cells of multipotent progenitor, expecting every single cell to make all DC subtypes, we did not find that was the case. So while 50% of the progenitors produced all three DCs, there were many clones of many cells, each clone, that only produced CDC1s or PDCs or CDC2s or combinations. So what's controlling this? We don't have a marker to separate them out. We couldn't find anything in single-cell RNA-seq data. And so we sort of have this Heisenbergian problem. So for those of you who remember, high school physics, the Heisenberg uncertainty principle says you can't know a particle's location and momentum at the same time because measurement of one precludes measurement of the other or knowledge of the other. And there's sort of a similar problem with single-cell RNA-seq. If you think about it, you've got a stem cell, you drop it in a well or inject it into a mouse and it makes only orange cells. And you're like, fantastic. Wouldn't it be, it made orange cells, but I don't know about what was special about its parental cell that gave rise to orange cells. Alternatively, you do single-cell RNA-seq, and because it's destructive, you know the transcriptome of that cell, but you've lost the opportunity to study its clonal fate. So there's two solutions to how you could link gene expression with fate. The first is a time machine. Once you establish that that single cell makes only orange cells, you can go back in time and measure the gene expression of that cell and then correlate it. And why not go back in time 3 times for triplicates and then do that not for 1 clone, but for 1,000 clones. And now you have all the gene expression correlates of lineage fate bias. You can link the two features together. All right, don't have a time machine. So the next best thing is you can let that cell divide a few times. And if you can demonstrate that the siblings of that clone are highly correlated in their fate, then you make a fair assumption that the programs that are controlling that fate are imprinted in that cell. So we did this for 108 clones, and we tracked their output as well as their RNA-seq profile in a technique we call cis-seq. You drop in a single cell, you let it grow to a small clone, then you split that clone. Technical replicates for DC generation in vitro. where you then assess the fate of those green cells by FACS. And at day 2.5, when you split them, you take a single cell, an RNA-seq or single cell RNA-seq snapshot of their siblings. So now you can link fate with gene expression. And we call this cis-seq because it's clone splitting in cystic conditions for parallel fate and RNA-seq assessment. And long story short, through the analysis of Liu Tan, who was a joint student with me and Matt Ritchie, he performed linear regression analysis to find those genes that correlate with fate. And so the way you read this ternary plot is that each dot is a gene, and that gene's location in the ternary plot is when it's enriched in clones that are biased towards PDC fate. So these genes are strongly associated with a PDC fate bias. These were the CDC2 fate bias, and these were the CDC1 fate bias. Importantly, if we just did standard differential expression analysis, we wouldn't see 450 out of these 500 genes, right? Because a lot of these regulators, a lot of these putative regulators that correlate with clonal fate look like noise and would be thrown out of your standard single cell RNA-seq analysis. But now we can link clonal fate, we can pull them out, and the noise is not noise, it's a feature. Okay, but correlation is not causation. So we took wild-type bone marrow, Cas9 transgenic bone marrow, we transduced them with a mini pool, 4 guide RNAs each of 500 genes, so 2,000 guide RNAs total. Cultured them, sorted the DC subtypes, and looked for changes in guide RNA frequency between the wild type and the Cas9. So if you get a reduction in a guide RNA in Cas9, it means you knocked out a regulator that was important for that cell's fate. Vice versa, if Cas9 is higher, you've knocked out a negative regulator of that cell's fate. So in this way, we identified 30 putative new regulators that are here on the right. Everything in green was an internal control we spiked in that we know affects DC development. Everything in orange was a gene we knew about, but we also discovered it by cis-seq and validated it with CRISPR. And black are all the novel genes we found. And we pursued this gene here called BCOR, which massively increased the number of CDC2s and PDCs when you knock it out. And that's demonstrated here. 4 independent guide RNAs have this phenotype here. And here are some of the other histograms of the cell numbers or the guide RNA frequencies, which is a proxy for cell number of different genes we found in our controls and in our data, novel hits. Okay, so we found the correlates and now we validated causation. But if you knock out a gene and cell numbers increase, you actually don't yet know what accounts for it on a clonal level. So this is a bit mind-bending, but bear with me. It's another time machine problem. So what is a clone's fate ordinarily? And if you could go back in time and then knock out the gene and then test what it would make, what is its fate in the absence of a gene? And wouldn't it be great to be able to compare per clone what a clone would do normally and in the presence of perturbation, gene knockout, cytokine, whatever. So it's another time machine problem. And so we invented cis-skew. All right, so how can clones be skewed in fate? They can either gain fate, they could either lose fate, they could either expand in number or they could contract in number. And these are not all mutually exclusive. It could be combinations of all of these and then cell type specific as well. So we came up with this experimental design called cis-skew. And I'll give you the simple version. Basically, you barcode your cells, you grow them up, and then you transduce them at 50% transduction efficiency. So half of the clonal siblings are knocked out for the gene and half of them are wild type, and they're cherry labeled. So now you can let them go, and then you sort the cherry positive DC subtypes, the cherry negative DC subtypes, and you compare the same barcodes to each other and ask, how did a clone's fate change in the absence of BCOR? And what we found, remember I told you that CDC2 numbers went up and PDC numbers went up, but not so much CDC1s. And so what we found was now in our heat map here, every row is a barcode, every column is a cell type, but these are the 3 wild type cell types, and these are the 3 BCOR knockout cell types. Here is an example of clones that in the absence of BCOR simply ramped up their cell production. They produced more cells. And here is an example of a cluster of clones that could not make plasmacytoids. They were destined not to make plasmacytoids. But when we knocked out BCOR, suddenly the ability to make plasmacytoids was released or revealed or activated, whatever you want to call it. And so now it was an example of fate gain. And in the paper, we show that the increase in CDC2 numbers is mostly explained by clonal expansion. And the increase in PDCs is a different mechanism. It's mostly responsible through fate gain in plasmacytoid fate. So to summarize this work, clonal multi-omics assesses daughters from one founder cell for different features. CISSEQ identifies the genes that control clonal fate. CISSQ identifies how perturbation affects clonal fate. And BCOR is a novel regulator of dendritic cell development. Okay, how am I going for time? Good. So take a breath. And then we'll ask about emergency development. So in the steady state, when everything's, ticking along, you might get hematopoietic output that makes B cells and dendritic cells. But then in an emergency situation, let's say a viral infection, you might have an increase in the endogenous levels of FLT3 ligand, which is the cytokine that promotes DC development, and you get this big expansion in DCs. And this is relevant clinically because FLT3 ligand is being injected into patients and preclinical models that demonstrate it can be added in conjunction with checkpoint inhibitors, and with activation, you can get enhanced cancer control. But if you think about it, how does a system make more cells? And there's three possibilities. One is clonal expansion. So as I just mentioned to you for the CDC2 fate, you have cells that were DC primed and they just make more of them. Now using barcoding, you'd have an increase in DCs, but no change in the number of barcodes and no change in the number of blue barcodes. And I'll get to that in a second, what the blue ones are. A second possibility is that you recruit cells into the process. So now FLT3 ligand levels bring in progenitors to contribute extra to the output of dendritic cells. And there you would expect to see an increase in barcode number. Now, some of these may be dormant progenitors that were not doing anything previously, or they might be progenitors that were otherwise destined to make B cells, but have switched fates to now make dendritic cells. And in that last scenario, you would expect an increase in barcodes for DCs, but also a corresponding decrease in barcodes for, let's say, B cell prime progenitors. And I won't show you the data now for the sake of time, but basically we found that it was clonal expansion that was the major contributor to emergency hematopoiesis. Now, that means that a clone is being instructed to ramp up its production of more cells. Now, the question is molecularly, how is this happening? So Dawn came up with this other technique called DiviSeq. So she CTV cell trace violet labeled LSKs, which are the hematopoietic stem progenitor cells. And CTV works such that when a cell divides, the fluorescence of that dye halves at each cell division. And so by FACS, you can see these peaks that correspond to cells at division 0, 1, 2, etc. So she used this to her advantage, where she could transplant CTV labeled cells, in this case, non-irradiated mice, inject the cohorts of mice with either PBS or FLT3 ligand, harvest the bone marrow, and then do index sorting into 384 well plates to do cell-seq. And not only does she have the CTV information, but also all the other classic stem and progenitor cell markers, and can now understand what's going on. And so she had this number of cells from each of the conditions. Now, this is what the data looks like by FACS, which is already quite enlightening. So what you can see in conditions of PBS, progenitors that are normally c-KIT high to begin with rapidly downregulate c-KIT and start to differentiate as they divide, as indicated by this arrow in this direction. CTV cells are dividing from right to left, and c-KIT is being downregulated. By contrast, in the presence of FLT3 ligand, they maintain c-KIT expression and CD34 and some other markers. So they maintain this sort of stem-like phenotype in the presence of FLT3 ligand to undergo more division and therefore presumably greater clonal expansion. And then they differentiate. And so there's this maintenance of a stem-like state over cell division in the presence of FLT3 ligand. So now here's the single cell RNA-seq data that comes from that. It's being clustered in an unbiased way into these 10 clusters. And I just want you to focus on cluster 9 for the sake of this talk. Here, we find that this cluster, most of the cells in this cluster belong to those in the FLT3 ligand condition. They tend to express high levels of c-KIT and SCAR1, c-KIT and CD34. They are mostly very rapidly divided, meaning they're CTV low. And they have a strong gene expression similarity to multipotent progenitors and common dendritic cell progenitors from ImGen data. And so when we looked at this data in other ways, so our hypothesis is that this is a DC biased stem progenitor cell. And so when you look at CTV and PBS versus FLT3, and now the y-axis here is an MPP score, you see this kind of population up here of a divided MPP-like cell. And when you look at cluster 9, they are all enriched in the top of that cluster. You can also look at the cells using a CDP score versus an MPP score. And again, there's this aggregation of cells in the FLT3 ligand condition that sit up there. And they are also, a lot of those are from cluster 9. And they have a DC progenitor, an early progenitor signature, a hyperproliferative history. And so what the final model is that when you have FLT3 ligand, you get program data I'm not showing today. IRF8 gets induced, ID2 is induced. So you prime the HSPCs towards a DC program, but you let it go a few more divisions. So now you can expand that pool of cells that contributes to the DC. And so this is what we call clonal tuning. And if I had to retitle the paper, I'd call it Emergency Hematopoiesis through Clonal Tuning. Through transcriptional remodeling of a cell, tunes that clone's fate to the destiny you need more of. In this case, we need more DCs in the presence of FLT3 ligand. The same could apply to with GCSF, with granulocytes, and so on and so forth. Okay, and that came out last year. You can read that paper. And the first author is Dawn Lin. She did all of this work, heroic student. She's now doing a postdoc at DKFZ with Andreas Trump. So finally, clonal cancer biology. All right, in this paper in 2018, we could separate clones into shedders and seeders, and I'll explain what that means in a second. As those of you who study cancer know, cancer generally arises from a single cell, and the thought is through a series of mutational events indicated with M here, it changes the program of the cells, and you get subclonal expansion. And these subclones vary with their ability to invade, metastasize, and resist treatment. Now, there's this sort of assumption in there that it's all genetic, it's not epigenetic. And the second assumption is that clonal frequency correlates with the ability to metastasize or resist drugs. And we found that was not the case. So this is a bubble plot. I'll just show you one data slide from here. Here are all of the clones in a triple negative breast cancer patient-derived xenograft. So we take the xenograft, we barcode label them, we put them into mice, we let them grow. When they grow to a certain size, we remove the tumor, and then we let the mouse continue to live until it develops metastatic disease. And now we can correlate the barcodes in the tumor at the time of resection to the barcodes that end up killing the mouse due to metastasis. And what you find, much to our surprise, is while there are some clones that are metastatic that were large clones in the primary tumor, like this yellow clone that's present in CTC, lung, and liver, there are other large clones you never see again, like these two purple clones up here. Never see them again. And by contrast, there are lots of small clones or clones that were below the limit of detection that you only find in the metastatic setting months later that were in CTC, lung, bone marrow, relapsed tumor, uterus, kidney. So what we found was a lot of these cells were shedding, a lot of these clones were shedding progeny into the circulation, but only a subset of them were seeders that ended up making the metastases of clones. So now our work is to say, I'll tell you what our work is in a second, but in this paper, we also said, well, what happens when you give chemotherapy? Now we have the ability to measure, do clones extinguish with chemotherapy? And, let's look at this data and say, well, if I give chemotherapy, at the two extremes, my data looks like this again, or my data looks like this, where there's just a few clones selected after chemotherapy. And here's the actual data from our paper: vehicle, cisplatin, 1 cycle and 2 cycles. And much to our surprise, there was not really significant difference in the number of clones that contributed to the relapse cancer. It wasn't like this, it was like this. So it tells you, and these were non-palpable tumors after chemotherapy. So it tells you that at least one cell from every clone survived the cisplatin. And once we took it off, they all grew back again. Now, for some of you, that will be no surprise. For some of you, that'll be a big surprise. I guess either way, the point is now we know the answer for this particular PDX, for this particular chemo. Could be different. All right, so now we want to know not just the patterns of clonal behavior, but the predictors. So now we bring in cis-seq, right? So here's the experiment that's ongoing. It's unpublished for now. We barcode triple negative breast cancer cells. We pre-expend the founders. Some siblings go for clone splitting into mice where we can identify which clone becomes lethal. And other ways we can find which genes correlate to the lethal clones through cis-seq. And we want to identify the regulators of metastatic capacity and drug resistance. And this is just preliminary data now, where if you do single cell RNA-seq of this MDA231 cell line and find this cluster 3, you would not know it's different to any other cluster if you just looked at it. And here's the heat map of that data. But now if you track which of the barcodes were present in which of the clusters, that ended up being the bulk of the tumor and the majority of the metastatic disease, they came from cluster 3. So we're trying to figure out what is special about cluster 3 that allows it to grow and metastasize. And we want to do this with MDA, which is a cell line, but also PDXs. And lastly, this paper came out late last year. This is a collaboration with Mark Dawson's lab, and Katie Fennell was our joint student. And her and Dane Vasiliadis, who are joint first authors, identified what are the predictors of lethal clones in AML. Heroic amount of work. And they identified SLIPI as one of those regulators, where if they knock it out, they can then increase the survival of mice transplanted with AML. And that came out in Nature last year. So you can see the power of clonal biology in many different areas. So what's next? I think I have 10 minutes. I can talk.
Shalin, you have plenty of time. Please go ahead.
Yeah, cool. No worries. So I hope I've given you a flavor of how you can use clonal information to understand patterns of behavior. And if you pair it with RNA-seq, you can find out interesting features of clonal behavior in immunology, development, cancer. But right now, all of these technologies involve lentivirus barcoding. So you need to pull out the cells, introduce the barcode through a lentivirus, and then inject them back into a mouse. But wouldn't it be cool if you could barcode the cells inside a live mouse without ever pulling a cell out of the body? Now, we're not the first to do this, but I'd like to present some unpublished data on this new tool we have that we think is going to revolutionize how we can do in vivo, in situ clonal lineage tracing. So this was really driven by Tom Weber. He conceived the idea and we discussed it at a meeting 10 years ago, I think. And he came up with it. He's a theoretical physicist by training. He had never touched a pipette before. He came up with this idea. We talked at a meeting. I said, come over to Australia, learn to pipette, make the mouse. So over the last 7 years, with a lot of help from other people, we've got to that stage now. We've got the mouse, we've got some data coming out, and I'd love to show you what that data looks like now. So the LoxCode mouse is where you have a series of DNA barcodes flanked by alternating LoxP sites, such that when you add Cre recombinase, those Cre sites, depending on their orientation, will either cut out a piece of DNA or flip a piece of DNA. And that can occur all throughout here. And would you believe those 13 barcodes can give you a theoretical diversity of over 30 billion different barcodes? In practical terms, you don't get 30 billion barcodes in one, obviously, individual experiment. That's impossible, but you get a lot of simple barcodes, easy to make barcodes that contribute to 90% of your data. You have to throw them out, but that 10% is complex and is super valuable to study lineage tracing. And here's what the recombination kinetics look like 1, 3, and 6 hours after tamoxifen addition in vitro. And Denise contributed to building this mouse. You might have heard of Polylox, which is this great tool that Hans Reimer Rodewald developed, and identical in principle. right? And they were independently conceived. Tom wrote his theoretical paper before this one came out, but yeah, we just didn't have a chance to make it in time. But this paper is great. And so it was the first demonstration of it. And with this 9-element polylox barcode, they can generate 1.8 million barcodes, and that you need 2.5 mg of tamoxifen, and you need to sequence with PacBio long read because they're long elements. In our case, the short barcodes, we have 13 of them. So we have 16,000 times more barcodes. We can use a 50-fold lower dose of tamoxifen. So for those who are worried about toxicity, and because we can sequence them on Illumina, we can get 100 times more reads and samples processed. So that's hugely beneficial from a very practical sense. And so our first proof of principle experiment, which turned out to be a huge project in itself, is mouse embryogenesis. So crash course, we all start from a single cell, and in mice at day 5, you have the epiblast, which is a ball, a bag of 200 cells. And there's a lot of debate about how much each of those contribute to all of the tissues that comes out in the day 12 embryo, where you now have brain, kidney, lung, liver, gonads, whatever. So do all 200 cells contribute equally, or is there some degree of lineage bias? Now, I'm not saying the lineage bias is intrinsically programmed. It could just be an artifact of where it's located in its 3D topography. We can discuss that later. But the question is there a lineage fate bias? Not a lineage, necessarily lineage priming, but a lineage fate bias of those single clones. So here's the experimental setup. You cross the LoxCode mouse to a Rosa-Cree RT2 mouse. And when the female is pregnant, 5.5 days after the plug, you give tamoxifen. Now you create barcodes in the epiblast, the 200 cells. And then those 200 cells divide and differentiate into the embryo. So you go from 200 to greater than 10 million cells. Okay. Then we can process this in 3 different ways. We can either chop this up into pieces of tissue and do bulk PCR for LoxCodes and compare LoxCode frequencies between tissues. We can do single cell RNA-seq and detect the LoxCode in the transcriptome because it's the LoxCode's under the Rosa locus, so it's transcribed. Or we can do spatial RNA-seq and look at the 3D architecture transcriptomically and clonally where the patches are all contributing. I'm not going to show this data today, but I'll talk briefly about these two. So this was a collaboration with Samir Taudi's lab in Weihai and with a very talented postdoc, Christine Bibbin. And so she dissected this little tiny 4mm embryo into 40 pieces, each one, 3 of them, into all of the tissues you can see here, plus a few more that you can't see because it's hard to show it on this map, and process them differently. And so Tom and Christine together did this heroic experiment where they dissected it, they lysed overnight, then they did LoxCode PCR and library prep of these 3 embryos, 288 samples, and sequenced them on a MiSeq. There was a lot of MiSeq runs as well. But we got the data, and this is the summary. So this is an intertissue clonal correlation analysis. So here are the tissues by their lineage definition, right? Blood, mesodermal organs, meso and endo organs, forelimbs, hind limbs, latent mesoderm, neuromesoderm, brain, neural tube, etc. along the top here. Here are the individual tissue types that belong to these macro tissue types that we dissected, but their ordering and the heat map is done in an unbiased way. So as you can see, there are tissues that are more clonally related to each other than other tissues. For example, this green patch of tissues are all of the different brain tissue that are highly correlated to each other, but lowly correlated to, let's say, kidney development or what's this, the forelimbs and hind limbs and the gut and stomach, right? And then here there's the forelimb buds and the hind limb buds, and they're more related to each other. Also related a little bit to the forelimb, left leg, hind limb, and right leg, but uncorrelated with the other ones. And so you can see this clonal correlation. Another way to look at it is in a UMAP. So now each dot is a tissue and its position is related to how clonally correlated they are with their neighbors. So these neural tissues are more correlated with each other. than these, limb tissues, the gut, liver, and organ gonads, these lateral mesoderms, and there's structure in that data. And yet another way to look at it is this like a clonal connectivity by tissue. So here we've just spread out the data points in a rough anatomical map. We've done that manually. But then Tom has found, it's like a nearest neighbor network analysis. And he's asked, for any given piece of tissue, what are its nearest neighbors in terms of clonal relationships? And he's done that for each piece of tissue to every other piece. And if you split that up now into tissue that you start the comparison with, you can see there's some really nice patterns. So neural tissue, both the brain and the neural tube, are more related to each other than the organs, which are interrelated with each other, and the gut that's related with each other, and the limbs that are related to each other. So there's all these tissue level clonal relationships that are a consequence of the epiblast having different lineage biases at a single cell level. And this has never been done before, like in vivo in this way. So I think, some of these results may not be surprising to the hardcore embryologists, but I think there's a first demonstration of what it actually looks like. Now we can track each clone, right? But I've just shown you tissue correlations for now, but now let's look at the individual clones. So here's a U map of the single cells. Each dot is a clone, and its collection in a cluster is by similar clonal fates. And so there's 6 clusters here, and these 6 clusters give rise to different tissues at different biomass. So cluster 6 is mostly giving rise to blood and organs, whereas cluster number 2 is mostly giving rise to brain and neural tube and other tissues, and so on and so forth. So there's lineage bias there. And now we can look at the individual LoxCodes. So here are all the LoxCodes on the x-axis and all of the different tissue types on the y-axis. And you can see the nature of the tissue bias of these clones is enriched for these tissues here. These clones are enriched for the brain and neural tube. These are the genuinely multi-outcome clones that are contributing to all tissues. These are also brain and neural tube, but a bit of lateral mesoderm. And there's another subset that are lower output, but more biased towards the limbs, and so on and so forth. So now we can track the epiblast fates to the tissues on a clone by clone level. Okay, that's all great, but it's all at a tissue level. But what about a single cell level? So on the same day, they also took some of the tissues because they digested them. They took some of the cells, and then they did single cell suspensions, multiplex them using hashtagging, using multi-seq. And I think we did 10-plex hashtagging, and then 4 different lanes so we could get the 40 tissues. Ran it on a 10x, and we got the cDNA and looked at gene expression, but also did a selective library amplification for the LoxCode and then sequenced that on a MiSeq with the cell barcode. So now we can correlate single cell RNA-seq with LoxCode identity, do that together. Here's the single cell RNA-seq data of the 40,000 cells we got across that day 12 embryo. And now, sorry about the resolution on this, but now, so each one of these is a different LoxCode. -11 is a complex barcode, 1 -6, 9, 1, 10, -5, -4, 13. These are all complex barcodes, and we selected clones that had a distribution that was not random, that was some form of enrichment. And so you can see these clones have an enrichment on this side of the cluster, these down here. These are genuinely spread throughout the mouse, and these have this peculiar enrichment here and here. And another way to look at that is then we group those transcriptional clusters by annotate. So these are the rows here are annotated transcriptional clusters that we've condensed for simplification. Along the bottom are the tissues from which we got those single cells. So bulk tissue, and then the rows of the transcriptional cluster within each of those tissues. And then the barcode, the number of cells with that barcode that were detectable across those transcriptional clusters and those tissues are then highlighted. So -11 is present like this, 1 -6, 9 is present like this, and so on and so forth for the different barcodes. And so sometimes what you see is the same transcriptional fate across tissues, like in this case here. And other times you see diverse transcriptional fate within a tissue. So there's the same tissue, but different transcriptional fates here, 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 and here. So, you know, we're just at the beginning here, and we're trying to improve the LoxCode detection efficiency a bit more. Right now it's between 15 and 25% detection efficiency. We'd love to get that higher. But this now allows you to link clonal fate with transcriptional heterogeneity in a native setting. And so, as you can imagine, we're applying this tool to the study of hematopoiesis, cancer, et cetera, et cetera. I'm going to finish up there, remind you what we work on and how clonal tools can help us do that. And I hope I've demonstrated to you the importance of integrating these three aspects. And these are all the people that have done the work. So just to highlight again, look, Daniela and Daniel really drove a lot of the single-cell RNA-seq development, and now in the single-cell core at WeHi. Sarah drove a lot of the B-core validation experiments that you saw, including SysQ, where Tom did the analysis. Tom conceived and did the LoxCode mouse. He was also involved in the breast cancer barcoding paper. Tom contributes to almost everything that happens in the lab. Dawn did the emergency hematopoiesis paper. Katie was in the Nature paper around AML clonal resistance. Louis helped with a lot of bioinformatics in the immunity paper. Antona did the breast cancer barcoding work, and I haven't been able to talk about the other students and people in the lab. But thank you very much for your attention. And here are I won't go through all the acknowledgements for the different projects, but thank you for your time, and happy to take any questions.
Thanks, Shalin. That was absolutely dazzling. It's an exciting field, and you've really shown us the explosive possibilities you have with all these technologies. So congratulations. I encourage everyone to put their questions in the Q&A, and unless you're a host and panelist, in which case you can't use the Q&A, you can type your questions in the chat, right? But for everyone else, please use the Q&A button. So lots of questions, but maybe I'll start with the very last one, okay, where you're looking at the fate of different transcriptomes in the mouse embryo. In that case, you know, the cell is either destroyed for single cell RNA-seq, or, you know, the embryo is left alive, and you look at the fate later on. So then, you know, is it possible that you have some stochasticity? You know, the transcriptomes, the clonal distribution you see in the UMAP of the single cell transcriptomes may be different from the clonal distribution in the embryo that actually developed and was profiled later. Is that possible?
If I rephrase your question, was the clonal fate bias an artifact of where it was positioned, and so it was destined to end up in brain? And was that imprinting happening much further downstream in the development process, or was the lineage bias already imprinted at the epiblast stage? Or have I got your question wrong?
So maybe I got the technology wrong. So my understanding was that you're doing the single cell RNA-seq at an earlier stage on one embryo. No,
sorry for that. Okay. I should have clarified. So while we do this cis-seq approach for all of the other projects, the central criteria for cis-seq to work is you need to be able to pull out clonal siblings soon after division to then correlate its fate later. You can't easily pull out clonal siblings from a developing embryo in utero. So in that particular case, we're only looking at the barcode inheritance in the destiny cells. We have no information about what its program looked like at day 5, only where that clone ended up at day 12. And so the single cell transcriptomics is of a day 12 mouse, and we're simply overlaying the cell identity based on its LoxCode identity and saying, oh, this clone didn't contribute to everything. It contributed to just these tissues.
Got it. So then all the profiling in that case is at a single time point, or all of your data.
Correct.
Okay, I understand.
Yeah. No, that's okay. I skipped over that very quickly, the experimental design. Yeah.
We are so happy we recorded your talk, so I'm going to dig through this again. Okay, there's one question in the Q&A, and then.
Sure.
All right, this is from Swain Chen, GIS. cis-seq and cis-q seem to require limited clonal expansion prior to splitting. And you showed that cells can differ in their maintenance cell state over a few divisions the cell state can maintain. Do you have any data on whether those initial expansion conditions might be contributing to the lineage biases you end up seeing?
Yeah, almost certainly environment dictates clonal fate. But what I can say is that all of the clones we studied were in identical conditions. And a lot of the clones, well, for the barcoding experiments, all the clones are together in one well. So the conditions are equal. But of course, if we change the cytokine concentration or change the media or something like that, it might affect the lineage priming. So instead, what we believe is, for the most part, the imprinting was already there in the mouse before we pulled it out of the mouse to barcode label it. And all we're doing is revealing the pre-existing lineage imprinting. Now, of course, cis-seq, I think of it more as a screen than a ground truth data set. You then have to go and validate each of these regulators that are correlated with clonal fate. So to answer your question, yes, initial expansion conditions can contribute, but for the differences we see, it is not contributing.
Thanks, Shalin. One more question from Swain that really appreciates the LoxCode technique, but then he's connecting it to problems in phylogenetics where we have homoplasy, right? So basically the same mutation appearing independently twice or 3 times. And then, so then how do you address that and maybe change your barcode design so that that happens less often?
Yeah, we would love to change our barcode design so that happens less often. So the answer is yes. What you're referring to as homoplasy, we refer to as simple barcodes. And so what Tom has done is he's got basically a matrix of simple and complex barcodes, and it's not a binary thing. It's you can scale complexity from 1 to whatever. And so we have a complexity cutoff of a certain number. And so we eject all of our homoplasy barcodes and only focus on the complex barcodes. And data I didn't show you today is between the 3 embryos, when we apply that filtering algorithm, there's only 1 barcode left in common amongst 200 between all 3 mice. So that tells us that we've been pretty good in getting rid of simple, non-informative homoplasy barcodes.
Right, and that'll help with richer data, more clones for the same amount of sequencing, I guess, right?
Yeah, look, it's like with any technology, you can't have everything. And so with this, unfortunately, we get a lot of useless barcodes, and we can't yet detect LoxCodes in every single cell. So for the first problem, I'd love to hear suggestions of how to reduce the chance of simple barcode generation. And for the second, we're trying different ways to increase capture efficiency.
Of course, yeah, it's just the first step. So,
yeah,
but still incredibly powerful, the number of combinations of recombinations you can have. Recombination combinations. Okay, can you compare how lineage tracing, the lineage tracing approach using CreLox, how it works relative to CRISPR-based methods? So that was one of my questions as well, is compare it to advantages and disadvantages of other lineage tracing CRISPR-based technologies.
Yeah, so for those who aren't familiar, there's several papers that have come out where they use sort of a template or a scratch pad, where then you have a Cas9 that's targeted to that site and creates random indels in that site, and those indels become unique barcodes. And so you can have different homology, for example, with the guide RNA and the scratch pad, so that some indels happen more frequently and some happen less, and so you get your complexity. Now, the power of those approaches is that you can get those mutations happening if not every division, every couple of divisions, so that when you get your single cells, you're able to reconstruct a pedigree. We can't do that. We can't reconstruct a pedigree, which kind of sucks, because you want to know when did it branch off to make cell type A? Did cell type A's trajectory branch off earlier than B and C and then B and C later? These sorts of questions. That's the power of CRISPR barcoding. The weakness is that the indels sometimes render the sequence kind of uninterpretable, or they just shred the whole barcode. You do need to make sure that your sequencing is accurate, so you're correctly calling your indels. You don't know what they are a priori, unlike LoxCode, so you can't. There's a filtering step that there's always a trade-off, right, between cutting off real barcodes and including spurious sequencing error-based barcodes. And I guess those are the main sort of pros and cons of the two approaches. I think you could probably now have, I can't remember now, is there a good CRISPR mouse that you cross to your favorite Cree? I'm not sure. But the point is, with this mouse, you can cross it to your favorite Cree right now and it'll work, sort of thing. There's a few caveats that I haven't highlighted now, but yeah, I think it's sort of more mature and more robust, but doesn't give you the pedigrees. I think that would be how I would say it.
That's true. Very interesting contrast between the capabilities. Lots of questions. I'm going to run through them quickly if I can. Sure. Do you have LoxCode mice and any other loci? I guess you just said you do, right? Not Rosa.
Yeah, no.
And how does the locus affect the function?
Yeah,
Lox function.
Look, the LoxCode is, I guess what you mean by function is like propensity for recombination, like in some cell types, is it closed up so it doesn't recombine, and other ones it's open. Look, to date, we haven't seen evidence of, like, we see this kind of homogeneous shift of LoxCodes in our PCR products. So we don't see a subset staying behind so that only some cells are recombined and others not. So I don't think that's a problem, the LoxCode in the Rosa locus. The transcription is not as high as we might like, so we might want to do that, add a different promoter in there. Steric hindrance, epigenetic effects, I don't think it's too bad. There's nothing, the embryologist who's working on this has not seen anything untoward. And keeping in mind that both tamoxifen-related toxicity and Rosa locus or putative Rosa locus integration effects can affect the biology, but she's not seen anything to date. And we've not seen anything in our hematopoietic studies that I haven't shown today either.
Thanks, Shalin. There's a sub-question. How about LoxCode in human tissues?
LoxCode, yeah, cell lines, sure. We'd love
organoids.
Organoids, yeah, great question. We do have a lentivirus that people are more than welcome to try out, but I reckon you're better off going with some of the other technologies that are out there now. I think Barbara Treutlein, I haven't read the paper yet, but Barbara Treutlein had a paper on this, I think, lineage tracing in organoids. But some of the CRISPR techniques might work. I just wonder if there are better techniques than LoxCode for human tissues. That's my instinct. We tried lentivirus for a bit. We wanted to do iPSCs. The problem was the LoxCodes are pretty unstable, so they just kind of spontaneously recombine, and then you have a recombined cell line, and then you're screwed. So, yeah, not so sure.
Thanks. There's a question from Jay Shin. He's asking about multiple recombination events in the same lineage. So what's the efficiency of free recombination? Does it occur every cell cycle, every end cell cycle? Have you tried stimulating with tamoxifen multiple times for multiple rounds of recombination?
Yeah, so the efficiency is super high because the LOX sites are so close to each other. We need a super low amount of, because we titrate the tamoxifen so we don't get not enough recombination and not too much recombination. You have to hit that sweet spot that gives you maximum diversity. So it's highly sensitive to recombination. All cells recombine in all the studies we've done to date. There might be some tissues that don't recombine well, I'm not sure. Have we done multiple pulse labeling? Yes, the problem is, while we would love to come up with a strategy where you only get flipping and not excising, we can't conceive of it. Because I think what you're implying, Jay, is what if you give a little pulse now, you get some barcodes, you wait a little while, and you give it another pulse, and you get some new barcodes related to the old barcodes. It just shreds. The LoxCode gets shredded over time till it's down to its simple barcodes. The other difference with PolyLox is we have 2,000 stable barcodes at the end of our maximum recombination, whereas they have just 9, the original elements. Ours we can get 3 barcodes such that the DNA doesn't bend far enough. But we thought about maybe a LoX mouse and an FRT mouse, and we put them 2 together, and then we could do one with one and one with the other. Yeah, I reckon, I don't know. Yeah, we're thinking about it. Not convinced that that's the way to go, but yeah.
Okay, well, certainly having multiple rounds would be interesting. We could somehow work. There's one more question from Swain, getting it again, something I was going to ask about as well, which is more complex scenarios such as stochasticity rather than deterministic expansion or deterministic fate bias or something like that. And so Swain's asking if there's potential for hard programmed timelines of development, as opposed to the opposite, which is an environmental responsive, perhaps epigenetic variable, stochastic model, and so on. And I'll add to that one more thing. How much purely technical variability do you have from replicate to replicate in these results for any of these effects that you showed?
Yeah. So there's definitely, let me just do a thought experiment. If a single cell can give rise to a clone of 2 cell types, at some point, there has to have been an asymmetry, right? Because at some point, whether that asymmetry happened right at the beginning of the pedigree and all of this trajectory was cell type A and all of its descendants, and this is all cell type B, or it goes all the way to the end and then it goes A, B, A, B, A, B, A, B, A, B, all along the line. So those are the extremes of when that bifurcation event happens. So there has to be a bifurcation of events, otherwise we'd all look like blobs. We just wouldn't have structure and cell types. There has to be an asymmetry, whether it's asymmetric cell division or some sort of stochastic asymmetric event, we don't know. I think that's the holy grail of tissue diversification. Okay, so we have to have an asymmetry at some point. So now let's go back to your question. In our data of CIS-seq in the paper, we analyzed 108 clones, and 98 of them were concordant in their fate, and the rest were not concordant in their fate. Now, for that paper, we focused on the concordant clones. But of course, the non-concordant clones are just as interesting, if not more interesting, because they're presumably either through a stochastic event or an asymmetric event, this clone's sibling went to cell type A and this one's went to cell type B. So all I can say is in that particular experiment, the majority of clones were concordant. If stochasticity played a role, it happened prior to the clonal expansion and clone splitting, not after. So I can't exclude what happened before, but I can exclude it after for those 98 clones. For the remaining 10 clones, who knows? Who knows? So I think, like all good things, there'll be nurture and nature in clonal decision-making. There'll be a lot more clonal memory. And so this is the term that I'm trying to inject into the field. There'll be a lot more clonal memory than people have given clones credit for, but there will obviously be an influence of environment that can change that. And so the whole CIS-skew style of experiment now, you can ask those questions. You could just split them in just equal conditions and ask how many times you see stochasticity, or you can perturb one of the experiment, ask how environment alters behavior. Does it shut down a clone? Does it reveal a clone? Does it skew a clone this way or that way? And so this gets now really exciting because you can understand how a SNP affects a genetic capability of a stem cell to hyperproliferate, how a cytokine influences clonal behavior, or how a gene influences fate decision-making. So these are all really good, big, hard questions that I think we can answer now.
Good to see that you're seeing some reproducible behavior across clones, and that kind of minimize. So if everything is stochastic, that's less interesting and
very interesting, but hard to tease apart.
And hard to say much about, perhaps. Yeah. But how about technical variability across replicates? Do you see much of that?
So it depends what
like
you mean.
if you do the experiment
Yeah.
on again and again.
Yeah, so if you mean technical variability in the patterns of lineage bias, we find there's we have variability culture to culture in terms of, let's say, DC generation. We might get more PDCs one time, more CDC1s the other time. The distribution of clone fate might be a bit different. But in that particular experiment, we pulled 4 independent clone splitting experiments together and then looked through linear regression analysis of those genes that correlated with that. So no doubt bigger numbers would reduce that. Now using lenti-barcoding would help give more confidence to more regulators, I think. So if you're talking about technical variability on fate side, yes. If you're talking about technical variability on the RNA-seq side, it's hard at this stage for me to give a figure on that because I guess let's call it biological variability is influencing the technical variability in that case from the point of view of RNA data. Don't have an answer to that question. But I guess, again, like I said, we use it as a screen so then we can then narrow down our list of genes to then go and target and validate, separately.
No, it was mainly the questions about the fate side, so you made major conclusions about fate and expansion and bias and so on.
Yeah.
How stable they are across, doing the experiment again. Right.
What I didn't mention, I didn't show the slide. We just track the output of the GFP cell, but the filler cells in the same well all have equal proportions of the same 3 cell types in every well in that experiment. They're all like identical, very tight variants. But the clones are like all over the place. So we've got this beautiful internal control for every clone we study.
Okay, that's again reassuring. That's fantastic. Yeah. I know we're over time, Shalin. We have a few more questions. Are you rushing to go somewhere else?
I'm good.
Okay, fantastic. We'll keep going for a few more minutes then. One more question from me, and then one from Swain. So so we've been asking lots of questions about on the technical side of these experiments, because these are new technologies and it's really fascinating to think about all the possibilities. I wanted to ask you about the biology of, say, your chemotherapy experiment, which was that surprising or not surprising? Of course, the thing is when you do something new, right, there's always people who tell you, oh, I knew that already. Oh, that's well known. But then when you look in the literature, there's actually no good evidence this way or that. And things that are within quotes well known are just biases and hunches, But the fact that the chemotherapy, you just presume that different clones have different clones. Let's say if you have a genetically complex or epigenetically complex cancer cell pool, you expect that some clones should be more susceptible and some clones should be more resistant for genetic or epigenetic reasons. But you didn't see that very much, right?
And so I was at pains to say in this model with this drug.
Sure.
So in that model, we were all surprised to see it from our perspective. When we talk about it, some people say, well, we know cisplatin just targets those cells active in cell cycles. So not all cells are active in cell cycles. So you only target those that are, and so you get shrinkage, but actually all the clones survived. No big surprise. So like you say, it depends who you ask. By contrast, in the AML paper with, oh God, what was the treatment now? I should know this. Sorry, I'm blank now. But if you look at figure 4 of that paper, you have a subset of clones that go from low to high frequency and another subset of clones that go from high to low frequency with chemo. So there you're getting a change and a shift. Now, they're looking at the dynamics of clone frequency change over time. The caveat of our breast cancer study is we're just looking at a snapshot between with and without treatment in 2 different mice. So was there a different ratiometric change in clonal frequency? Don't know. Probably there is. I don't think it's just a 1-for-1 that the big clones shrank and became big again and the small ones stayed small. I reckon there's some sort of mixing of frequency, ultimate frequency that happens. But we don't have a good way to do that in vivo. In vitro is easy. In vivo is a bit harder. But we're thinking of clone splitting in vivo and testing them in parallel to do that sort of, ask that sort of question.
Yeah, I think there's so many theories of what responds to chemotherapy, what metastasizes, and so on. I think this is the most powerful system for technology for really answering that question directly.
Yeah.
Admittedly, like you said, in specific model systems, but at least that's a start to test all the theories.
Hey, look, for most of us, we care about one cancer and one treatment, or finding one treatment to one cancer. So we don't have to answer all questions all the time.
Very much. Okay, one last question from Swain. Going back to the question of, asking for your opinion on the dichotomy between, say, very clock-like or completely programmed differentiation versus internally programmed differentiation versus external signal-based differentiation.
Okay, so I wanted to, but there's too much I wanted to talk about today. So please go read our Cell Reports paper from 2018, where we track the clonal kinetics of DC development in FLT3 culture. So with cis-seq, we just RNA-seq at time point 1, fate at time 2, and that's it. In the Cell Reports paper, there's no RNA-seq, but we track clonal fate over time. And now we've seen this not only in, I haven't told you what we found yet, but I'll tell you in a sec. We found the same pattern in hematopoiesis, in cancer, in another cell line, and so have other people. And I think this is going to be the big thing that happens now in biology. What we find is a lot of progenitors in the clone splitting experiment, we fly clones that divided enough to be split into 2 wells, and then just kind of ticked over through multiple serial samplings. And then the siblings decided at day 10 simultaneously to start proliferating. And other clones were proliferating and decided to start dying at day 8. And it was all clone intrinsic in that the cells in this well behaved exactly the same as cells in that well. And we see this with other cell lines too, that these quiescent clones, the siblings were in 4 different flasks, and 3 months later of culture, those clones simultaneously started taking over the culture. So I think there is a clock that cells have to determine their clonal kinetics. And I think that'll be the next big thing that our lab works on, is the regulators of clonal kinetics: size, timing, and lineage bias, and how that's perturbed with drugs, environment, etc. Watch
Fantastic.
This.
Yes, well, so that also answers the "what are you going to do next" question. So very good. And yeah, I know we pushed you over the time slot, but obviously so much to talk about here. I think it's a fantastic overview of what lineage tracing can do for you to answer very, very basic biological questions. So we will be watching this space, and we hope to have you with us again for another round. Thank you so much, Shalin.
Wonderful. Thanks so much, Shyam. Thanks everyone for listening.
All right.
Bye.
Bye-bye.